Hello to everyone. Thank you for the invitation in presenting our latest work. My name is Gabriele Ellena, and I'm currently working at the Belgian Nuclear Research Center in collaboration with the University of Antwerp, and I'm funded by the FWO. And I'm here today to give you an overview of our latest work exploring extremotolerant microorganisms to be used as supplements in deep space exploration. So here you have a picture of the Earth taken from the cupola of the ISS, the International Space Station. And well, not many people for sure have seen the Earth from this perspective, live, I mean. Um, but even though it's outside everything we know, even though it's a point of view completely different from our knowledge, we are still pretty close to the Earth. We are still 3-4 hours away from the Earth. Our astronauts are there. We ship them food. We ship them resupply, whatever they need. Here you have other two pictures actually taken from around the moon, from the moon and around the moon. One is quite old, it's from 1968, from the Apollo 8 missions. It's an iconic picture. The other one is actually only a few months old from Artemis 2 mission, and was taken in April. You see both standing pictures. You see the Earth. Here it's indeed farther from the previous one, but still actually quite reachable. We are talking about 3-4 days of travel. It's feasible. We can still be able to ship things there and resupply our astronauts with resources in case of needs. So, so a different story is for this picture, which was actually taken from Mars. You can see, actually, no, you cannot see, you can barely see the arrow indicating where Earth is. It's definitely more farther away. It takes around 8 months to get there. We cannot ship things whenever we want. We have specific windows. And for this reason, on Mars, we would need to be able to be independent, to be not Earth dependent for food and resource production as we are actually for the ISS and as we would be for the moon. One of the main way in which we can be independent on Mars is that the astronauts are able to produce their own food that can actually fulfill the requirements that they have on a planet that we don't know for human life and that is currently exposed to radiation. The main constraints for the food on Mars is that we cannot ship it, as I already said, whenever we need. But it's also the fact that we cannot store it. So we cannot send all the food we need the first time and then store it until the astronauts will need it because it degrades. It degrades already in normal storage even more under radiation. And currently there is no um, adequate acceptability of food that can be stored for 5 years. So, what was proposed now quite many years ago was the introduction of bioregenerative life support systems. So, systems that actually provide fresh food with and air and water with the recycling of the waste of the astronauts. The Melissa project from the Melissa community and partnering with the European Space Agency is one of these bioregenerative life support systems, which is aiming to recycle all the waste for the production of food, water, and oxygen based on what actually is happening in lakes currently. Um, But there is a but to these systems is the fact that currently we are still planning on providing and giving the astronauts only plant-based food, and actually a very specific and extreme plant-based diet because of many constraints we have in space, energy constraints, size constraints. We cannot grow all the plants we want and all the food the astronauts would need. So definitely additional sources for complete proteins and vitamins are needed. And to do this, an option would be using microorganisms. You see already here in compartment 4A in the graphic to your right, Limnospira is currently planned to be used both for oxygen production but also for biomass production. So it's edible and has all the essential amino acids in it, which is not that common in plants. And it's actually a good source also of antioxidants. However, the concept of this work was to actually improve these bioregenerative life support systems, plant-based life support systems, with more microorganisms. You see here an image of bioreactors to the right that would supplement what you see in the greenhouse, bioreactors to the left, sorry, that would actually supplement what you see in the greenhouse of to your right. Not only proteins, but also micronutrients, vitamins that are missing from the diet of the bioregenerative life support systems. So the way in which the work was planned was through several steps, starting from identifying the micronutrients and nutritional gaps in this plant-based diet, BLSS. And to do this, we developed a model, calculation model called Novae. We started from 11 species that we know are currently planned to be used in the Melissa systems. And what we wanted this model to do, here you see the QR code is actually published work. What we wanted this model to do is to reduce as much as possible the difference between the amount of micronutrients provided from the best combination of these 11 species and the requirements of the astronauts. And we did it by minimizing this equation. You see shifting over to 37 micronutrients, I, and the 11 different food species, J, for X, so the amount in gram of J, so the 11 different species, which was a variable, and the fixed value M, which is the amount of micronutrient I within the food species J, over the requirements of the micronutrient I. So we wanted to close as much as possible this gap. Then we had some constraints. For example, the amount of energy had to be between certain values, also the proteins, and we also didn't want to exceed the limits for the micronutrients suggested by the World Health Organization. So what we did before getting there, we went through several steps. We started from the first run of the mathematical model, Nova 1. What we did was, as mentioned, having some harsh constraints on the energy. So we wanted that the daily energy requirements would be between 2,800 calories and 3,150 calories. And at the same time, we also wanted that the protein requirements would be between the 10 and 25% of the total energy intake. And we didn't apply any other constraints. This was our first run. What we got was actually that we had three micronutrients that were excessively supplied, 21 that were correctly supplied, and 13 that were not enough supplied from the diet. Here in red in the right graph you see what was exceeding the limits, and in the left part you see what was not reaching the limits. So we have cobalamin, vitamin D, omega-3, omega-6, we have riboflavin, vitamin A, and few other inorganic micronutrients. So then we decided to improve and to add all the limits also, all the limits of the micronutrients, limits recommended by the World Health Organization. And spoiler, there was no possible solution to this. So there was no way in which, with these 11 species, we could provide all the micronutrients the astronauts need without exceeding the limits and staying within the specific amount of energy and proteins requirement. So what we did then was to introduce one limit, one micronutrient limit at the time, and see how many we wouldn't be able to stay below. And here what we got was only, well, one micronutrient that we were not able to respect. And you see it's here to the right, it's folate, which is actually exceeding of 331% the daily intake requirement and 132% of the limit. So then we decided to go for the third version of the model, Nova 3, in which we said, okay, having such harsh constraints on protein and energy requirement, it's not the focus right now. Because the energy requirements will change, will be highly dependent on the amount of exercise the astronauts will need on Mars, on the physiology of the body of the astronauts that will be chosen, their basal metabolisms. So we are still not able to assess an amount of energy that our astronauts will need. But what we are able to do is to know the micronutrients they will need, because for that we have fixed values spread it across all the population, independently from their BMI, independently from their basal metabolisms, their weight, their height, everything. So what we decided to do was to actually use the daily energy requirements and protein requirements as fixed values, not fixed, but yeah, as fixed values, and we wanted to get as close as possible to these values. So we took 2,975 and we told Nova, okay, get as close as possible to this number, but you don't have to stay within a very specific and narrow range. And also to the amount of proteins, we said, okay, try to get as close as possible to the 15% of the total energy intake from the proteins, but you do not have to be between 10 and 25. And then all the micronutrients limit, we applied them immediately. And what we got was a solution that was not exceeding any kind of limits regarding the micronutrients, but we got 27 micronutrients that were lacking, that were not supplied enough. Many of them were still above 80%, so that's good, but some of them, also seen in the previous models, were not supplied enough, like cobalamin, 0%, vitamin D, omega-3, omega-6, again riboflavin, and some inorganic micronutrients as well. Here there is just a representation of the limits for organic micronutrients and mineral micronutrients. As you see, in the first model we had the blue line, we had three different limits exceeding, so folate, magnesium, and phosphorus. In the second one, only folate, and third one, none of them, because all the limits were respected. So then here, after understanding which micronutrients were actually missing and were of most interest to be supplemented, what we decided to do was to identify and select the microbial sources that are able to give these micronutrients. And to do this, we decided to start with the largest database that we know, KEGG, and for metabolic analysis, and we um, collected all the microorganisms that have been annotated on the database for the production pathway of at least one of these micronutrients. So what we did here, this part is still on the same paper, same work that was published online. What we did was developing a new pipeline called Genomars, and um, we decided to rank the micronutrients based on how much they were lacking in the diet. For example, B12 was lacking 100%, was not supplied, so I had a higher priority than lysine, which was provided for 90%, had a lower priority. And then what we did was based on the annotation on keg of the microorganisms that were producing these micronutrients. They also got ranked which micronutrients were they producing. They got ranked also for previous space flights, previous simulated microgravity experiments, irradiation resistant experiments, their capacity of growing using in situ resources of Mars, and their metabolic flexibility. Are they strictly aerobic, anaerobic? Can they do both? Grow in both conditions, and radioprotector production, health promotion properties. Then what we did was also filtering them out based on the ones that are extremotolerant. So can they tolerate a wider range of temperature, for example, pH? Do we need to have specific condition to grow them, or they are happy even if we do not control temperature at 37 degrees or at extremely low temperatures? Yeah. And then we got a ranking, you can see to the right, where we started growing in the lab the first four, so the Brevichia sanseni, Bacillus subtilis, Lactobacillus reuteri, and Limnospira indica for the production of um, lipids and ergocalciferol, riboflavin, and cobalamin. Here there is also an example with vitamin B12 of what we wanted to do was to, well, B12 is lacking, has a radioprotective effect, it degrades after 1 year of storage, and is not provided. We saw it in the model. It's not provided in the BLSS, and there is a need for the astronauts of B12. But what we can do was to introduce it with Lactobacillus reuteri, which actually could provide quite a high amount of B12, and with 11 milliliter of culture per day per person, we would reach the daily intake recommendations. Okay. So from here, what we had to do was to actually test the robustness and the nutritional value under the space condition, irradiation, and simulated microgravity for these four strains that we are using. And actually we were able to test also space flight, so we had the opportunity of sending bioreactors with Limnospira indica inside, and we were able to show that there was no difference between the space bioreactors and ground-based bioreactors in micronutrients production. We also irradiated our culture of reuteri to see if the amount of cobalamin was different. What we see, and riboflavin as well, what we see is actually lower production of riboflavin in the irradiated cultures, not intracellular but extracellular. So it looks like secreted less. So then we did on the Baremicia Sanseni also some simulated gravity experiments, both Martian and low Earth orbits. And what we saw is that actually there is a difference with the control that we are using. So based on we're using some rotary cell wall vessels, and they have a membrane for gas exchange only on one side. So based on the side on which we are turning it, there is sedimentation of the strains close or far from the membrane, and it's reducing the boundary layer that we have in the simulated microgravity, leading to a higher oxygen exchange or lower oxygen exchange, gas exchange in general. So it grows faster indeed in one position of the control compared to the other one. Um, and the next step is to actually optimize the production using in situ resources. So what we have on Mars to do not have to only recycle, but being able to use what the planet also offers us. And I wanted to, well, acknowledge all my team for the work. So both the microbiology team, my mentors Felice and Natalie, the Bees and BR1 team in SCK, which allowed me to enter in the facilities, University of Antwerp for my promoters, and external members that I'm collaborating with. And of course, I wanted to thank you for listening, for inviting me, and I hope here there is my Linktree QR code. I hope you will be updated on I will be able to update you there on the future outcomes of this research. And I hope you enjoyed the presentation and the conference.